Hello, my name is Mike MacCoss, and I'm going to tell you a little bit about work that's being done in our lab about improving the robustness and reproducibility of quantitative proteomics. It's definitely an area that we've been working a lot on, and it's something that's definitely not a solved problem, and it's a work in progress. I think we're definitely getting there, and I'll tell you a little bit about how we're doing this and what the current state of the art is and where we still need to go. So if you look at pretty much how proteomics has been done classically, it's this methodology known as shotgun proteomics. And basically what we do is we take a very complicated protein mixture, we subject it to some sort of proteolysis, and then we get these peptides or smaller portions of proteins out, which have a much less diverse physiochemical properties than the intact proteins. We separate them based on some sort of chromatography, most traditionally microcapillary LC, where at each point in time we collect a mass spectrum. So on the x-axis is mass to charge and abundance. So we basically have a three-dimensional piece of data, and we tell the instrument, or the instrument makes decisions on the fly using a process we call data-dependent acquisition, where we select individual signals that are coming off the column and subject them to tandem mass spectrometry, or MSMS. So we perform multiple stages of this, and then we repeat the cycle over and over again. Modern instruments are getting extremely fast, so we can now collect data at around about 20 hertz or 20 MSMS spectra a second. That's particularly powerful and something that hardware-wise, this has been an enormous feat of engineering to overcome and perform. Okay, so why do we want these fragmentation data? So if we look at this data, we collect a mass spectrum and multiple MSMS spectra, and we have this individual peptide sequence, and we have an MSMS spectrum for this peptide sequence. We get a spectrum that looks like this, and the reason why it's valuable is because for an individual peptide sequence, we have two sets of ions that form between each individual amino acid. So for instance, here, and the difference between two adjacent amino acids is the mass of that side chain of the amino acid. So for instance, if you look between this bond break of leucine and phenylalanine, we get two different sets of ions, and one would be this 467, which contains the N terminus, and the other one is this 792, which contains the carboxy terminus, and so on and so forth. We look at the difference between this histidine and leucine. We have a mass fragment that's 353, which contains the N terminus, and 905, which contains the carboxy terminus. So we can use this as a unique signature of the peptide sequence, or we can just look at the differences between masses to know that this difference here is giving rise to basically a leucine amino acid. Back in 1994, Jimmy Yang and John Yates, this is 20 years ago now, came up with an incredible invention which led to the development of a software tool called seqUest, where you basically take these MSMS spectra in a protein sequence database, and you take each spectrum, which has a given precursor mass. Using that precursor mass, you go through all possible combinations of amino acids that would add up to a given sequence mass with some tolerance. You then, from that amino acid sequence, generate a theoretical spectrum based on the predicted fragment ions from the carboxy terminus and the amino terminus. And then you would try to match the two together and calculate a score. And the primary score that was originally done was a score known as the cross-correlation score. And this match is what we often call a peptide spectrum match. And then we use this to identify individual peptides. So we can use an approach by where we take intact proteins, digest them to peptides, and then we have these peptides, we subject them to fragmentation spectra, and then we can map these fragmentation spectra back to protein sequences predicted by a genome to determine the identity of peptides that are coming off the HPLC column. Now, there are some challenges with this, and that's what I'll talk about for the rest of the talk. And the main thing is that these mixtures are very complicated. So if you're to look at this data like such, and make a plot just of the MS scans, there are, and each of these little black dots in this slide is literally a peptide that's coming off the column, being plotted versus time and abundance, and intensity is being done in a grayscale. It's kind of coming out of the screen. So there are hundreds, if not thousands, of individual peptides coming up the column. And if you actually look at this a slightly different way, we now know that we can detect between 100,000 and 200,000 peptide features in an LCMS run. This is a lot. This is a lot of data that's coming off in a very short period of time. And most of our runs are between 1 and 2 hours. And so being able to make a mass spectrometer that can actually select and acquire data for 100,000 to 200,000 peptides that are coming off in a chromatographic timescale is still a daunting challenge. So just to kind of look at this a different way, if you took a slice through the data like so, you could plot the cross-section through this data. So this is signals. Here's retention time on the x-axis and intensity on the y-axis for things at 850, so things at a single mass to charge. There are easily hundreds, if not thousands, of things that have the same mass. And you can actually also plot it the other way, where you take a slice through data like such, and this is what we call a mass spectrum. So it says a mass to charge on the x-axis and abundance on the y-axis. And now there's hundreds, if not thousands, of things at in each spectrum at each point in time. So again, so modern instruments run at 20 hertz, 20 ms spectra a second. I've already told you that we can collect, we can see in a short period of time between 100 and 200,000 peptides in a run. But these instruments can only collect data on the speed of about 60 to 72,000 spectra per hour. So we're still never going to be able to catch up, at least not in the near future, to this sampling everything that's coming off on a chromatographic timescale. And again, I want to further emphasize that this is kind of a strange way of doing this, because what we do is, as we see things coming off the column, we're focusing our time and our energy on the most intense molecular species coming off the column first, and then we work our way down to the least intense ones later on in the run. To give you a sense of how crazy this is, and I guess this is the way our field's been doing this for 20 plus years, is imagine going and visiting a foreign city. So I'll give as an example a city like Kyoto in Japan. And let's say you were told you could only visit and explore the city by visiting the tallest buildings or tallest structures first. So you go to Kyoto, you go to the tallest building, it'd be some sort of office building or hotel structure, and you look inside of it, and it look very similar to a lot of Western cities, a lot of American or European cities. And you maybe look around and it'll look, looks very similar. There's no difference. There's a phone, there's a television, etc. You look out the window and you'd see, well, the city's, it's surrounded by hills. It's got this high-speed train. That's a little different. We don't have many of those in the United States. You look and see, well, they drive on the left-hand side of the road. But in general, things don't look that different from a lot of other cities that are in the United States or Western Europe. So if you so you'd miss a lot of really spectacular things about visiting Kyoto. You'd miss things like, the temples and the shrines and the storefronts and here's again another shrine and you'd miss the people and the different types of. You wouldn't experience anything about the culture of the food. You would just, it'd take you a long time in order to even get down to street level to begin to see these things. And of course, the stuff that's at street level is really what makes a city a very spectacular place. So, so if you wouldn't learn much about Kyoto by going to the tallest structures first, why would you use this approach to understand protein mixtures? And this is basically the question that our lab's been trying to address for about the last 10 years. Our methods have largely been used and based around using this data-dependent acquisition, where we focus collecting data on the most intense peptide features first, and then we slowly go down to lower and lower abundance ones. And even though instruments have been getting faster and we make it down to street level occasionally, we still spend a majority of our instrument time collecting data on the most intense features. So what is the general strategy that we should take, or we generally use? So if you go into a foreign city, what do we generally do? Well, often we build on other people's knowledge, right? You go and buy a guidebook, or you go and look in Google and you do a Google search. You say, okay, Kyoto places to visit, and And in there, you're already starting off at a much better place than starting at the tallest structures first, which is a very arbitrary decision to start at taller structures and go down to lower structures. And while it may not pick up all the best things and the most important things of going and visiting a foreign city, it's definitely going to be much better and much worth your time than starting randomly and just going for the tallest structures. So that's basically what we end up doing now, is we're advocating to people is that you start with things, knowledge about a given sample and disease type. We know a lot about biology already, and let's start to go and ask very hypothesis-driven questions as opposed to starting off just trying to fish and try to find things by hoping that the most intense buildings provide you a sense of the city. And again, you can look at things like Kyoto food. Should there be certain type of food or cultural things I should experience while I'm visiting there? Another example of why this isn't very useful, when I started the lab is now about 11 years ago, we had made a decision that we were going to use C. elegans as a model in vivo system for developing technologies. There were a lot of reasons why we wanted to do this, but some of the main reasons were it was the only multicellular organism with a genome that was sequenced to completeness at the time. And what I mean by completeness, meaning all chromosomes from telomere to telomere were sequenced without any gaps whatsoever. It was a huge tour de force and a major monumental effort in order to make sure that it was truly sequenced to completeness. And it was also the only organism for which we knew the complete lineage for all the 959 somatic cells. So, of course, John Sulston received the Nobel Prize for this work. So if you're trying to understand a system to completeness, one of the things that we obviously are missing still now is information about the protein expression and protein abundances within the system. We already know the genome to completeness, which was unusual, and it still is unusual to have a genome sequence to completeness. We also knew a lot about how this genome encoded for different lineage, but how this genome gives rise to a living functional organism like a worm is still something we had very little knowledge about. So we started off by using this also as a model system for insulin signaling, and of course there's obvious reasons for that, but the pathway, the insulin signaling pathway is highly conserved. So if you're looking at humans, the insulin receptor is encoded by DAF2, and PI3 kinases are encoded by AGE1, and PDK, and the FOXC4-KIT transcription factors are encoded by DAF16. So there's a lot of, by understanding and characterizing the signaling pathway in proteomics, using proteomics technologies, we felt like could help us inform some information about studying the same pathway in humans. So we decided that we would just take a brute force approach. We're pretty good protein biochemists, so we were going to fractionate proteins and with the attempt to improve our chances of identifying these proteins in a complex mixture. But we're trying to improve our identification using data-dependent acquisition. And what I mean by mud pits here is we took reverse phase the intact proteins, and then we digested the peptides and ran 10 multidimensional chromatography separations to separate the peptides. We did separate based on charge into 8 fractions, and then digested those and measured them by the peptides. And then we also tried to separate the insoluble proteins by density centrifugation and measure those. And each of these mud pits, these LC-LC runs, were taking 24 hours. So this is about 30 days of mass spectrometer time just for one sample. But again, we were kind of ambitious. We felt like we could work really hard and we'd be able to solve these problems. The big thing that we weren't expecting at all was that after collecting all this data, which was a ton of data, about 6 million MSM spectra, this is on older technology, and identifying a decent number of proteins, we identified 0 proteins that were known to be involved in the insulin IGF-1 signaling pathway. So again, we hadn't made it down to street level yet. We were just sampling the most intense features that were in the organism, most intense 6,000 or 7,000 proteins, and a lot of the proteins that were interesting to us in trying to understand this biochemical pathway were things that we weren't even beginning to characterize. So what are our biggest challenges? So the methodology was kind of locked in. Almost everybody was collecting data in this way. And in many ways, you basically have to take a step backwards before you can go forward. So we began to make some decisions and saying that, okay, well, we actually think it's this data-dependent acquisition that's starting from the top, the most intense structures and going down, or the most intense peptides working down, is what's causing our biggest challenges. And it also creates data that's kind of semi-random, it's irreproducible, and we can say stuff about what we detect, but we can't really say anything about what we're not detecting. And it's, again, we really struggled to do much of any type of quantitative analysis, which of course what we were really interested in. So that began to get us thinking about what we're now calling, and what the field now calls, targeted proteomics. We're using nano flow liquid chromatography, selective reaction monitoring, mass spectrometry. And this is a little bit different. Instead of trying to now sample everything that's in a mixture, what we wanted to do is start with a hypothesis. We want to basically do that Google search first, or look in that travel guide first, and start with these are the most important proteins for us to be able to measure, and then to be able to target, specifically measure those. And ideally, we'd be able to do this for hundreds of proteins in parallel. And that's where we began to get started. So we use a different type of mass spectrometer called a triple quadrupole, where we basically start with peptide sequences. We tell the quad to measure individual peptides. We subject them to collisions, and then we measure fragments in a second filter called Q3. And this becomes then a very selective filter. So basically, if you wanted to measure this peptide, we would measure that in these series of fragments, and then you'd have this chromatogram versus time, which is only reflective of signals from that precursor mass and those fragment ion masses. So this becomes a fairly unique signature. And we began to think of these things as kind of a mass spec Western blot. And the fact that these covary versus time means that we actually have encoded most of these information of this sequence backbone. And so that not only are we getting a quantitative measure of their signal intensity, but we're also getting a lot of qualitative confirmation that we actually are measuring what we think we're measuring. And so if you go back to the data that we were looking at before, remember we were looking at the DAF16 was the focal transcription factor, AKT1 was the serine 3 kinase in the insulin signaling pathway. And previously, we weren't able to detect them even with 30 days of mass spectrometry time. But now that we have a targeted measurement, we can use synthetic peptide standards, and we can now confirm in less than 25 minutes of instrument time that we can actually detect and determine the identities of these individual peptides, and we can have some sort of basic quantitative measure of the peptides versus development, versus condition, versus et cetera. So this is pretty exciting to us. This was pretty much a game changer. We now knew that if there's no shortage of hypotheses. We just now need to be able to go and rapidly test these hypotheses without having to generate antibodies for each individual target protein. The other thing that's pretty powerful in this is that the quantitative linearity is pretty much unmatched by any other analytical technology. So if you're looking at this individual peptide from this yeast protein that spiked into human plasma, and we made a dilution curve, and if you keep blowing up the bottom end of this dilution curve, it's still linear. Keep blowing up again, it comes off the curve a little bit, but it's still extremely linear. But basically, we have quantitative linearity for 4 orders of magnitude, 4 logs, and modern instruments now can do almost 5 logs. And the sensitivity is down to the mid to low atomole level, depending on the peptide. So here's a case where it says 12 atomoles, that peptide on column, in 10 micrograms, 1 fraction of human plasma, and we're able to still detect it. And these other peaks on the side, it's sort of like an antibody Western blot that's being done at very low abundances. You're going to get some sort of lack of specificity when you're trying to push the limits of sensitivity. But still, this is at the bottom end of what we detected. Okay, so I've sort of told you a little bit about the positive things that in 2006, when we started to do these experiments, we really struggled with the software. There was no way easily to start to tell the instrument to measure specific peptides for a protein, or which masses for that individual peptide. We had, while we could do that manually, it took a lot of effort. So we couldn't generate methods, we couldn't really look at the data, and we definitely struggled with being able to do this between instrument platforms. Each vendor sort of had their own tools. It was very hard to look at the same data between different platforms. So at that point in time, we hired a software engineer. We built a software tool called Skyline. This has been really eye-opening to me, this project. We started this vision between Brendan McLean and I, the development manager, with a couple key attributes that we felt like really needed to be in the software tool. One is that we wanted to be protein and peptide centric. Most software tools at that point in time were what I would call mass centric. So with a mass spectrometer, you tell the mass spectrometer to measure certain masses, but we didn't want to measure, mass 652 and 933. We wanted to measure this gene product with this peptide, et cetera, with this fragment ion. Other tools that were out there were either command line based, so they weren't that user friendly, or they were used as like a web browser type interface, and that also wasn't ideal because we wanted something that would sit on the instrument platform. We wanted the ability to do full undo, because if you're setting up a method to measure a specific set of peptides, you could actually imagine where you get to the point where you have a very large list of peptides, hundreds or thousands of peptides, and then you accidentally delete a peptide or a transition, and then you want to make it easy to be able to go back and add them in if you realize it was a mistake. So this is something that isn't appreciated that much scientifically, but as far as engineering-wise, was an enormous challenge, and we're still probably between academic and also commercial tools, we're the only tool of this type that actually implements full undo redo support. This is an open source academic project. We support lots of different, our view of targeted proteomics has changed. So we originally talked about SRM, but we've also expanded this to work out some more modern analysis strategies too, that aren't just specific to triple quadruple mass spectrometers like I had already pointed out. There's been a lot of talk within our field about sort of standards, standard formats. They tend to be a little cumbersome. They're not as efficient as a vendor file formats. Also, there's always some lost in translation, loss of information that's lost as you go from the vendor format to a standard format. So we wanted to make sure that we could read the vendor data directly without file conversion, and that was actually quite a bureaucratic hurdle to overcome because the vendors weren't at the initial time trying to make it easy for us to get data out of their software. We wanted it to be free and open source. Free because we wanted to do this as a service to the community. Open source because we wanted to just make it completely available to people to check our code, to make sure that we were doing what we were saying we were doing, and we wanted it developed professionally. And what I mean by professionally is not just that it was developed by true software engineers. We wanted it to use very professional software development practices as far as testing goes, code reviews, even the way we support the software as far as a support board and returning and responding to comments and criticisms and providing feedback and fixing bugs. We wanted this to be a tool that was truly supported and not something that was just developed for a project and then forgotten about. So one of the first things we did was to begin to look at this data. And one of the things I pointed out beforehand is there was no real tools for looking at these things across vendor types. This is one of the first times we started to display the data. So we took data from Manny Polovich's lab at the Fred Hutch Cancer Research Institute here at Seattle, which was an ABSciX instrument. And we had data from our lab. And we were able to plot it for the first time. This is the data from the Q-Trap, and this is data from our triple quad instrument. And you can notice that there are some slight differences in the data. We were able to look now for the first time ever at the same different peptides between different instrument platforms. We were able to use the same signal processing tools to be able to get quantitative data from both of them. This was really a game changer, and it was shocking to us that no one had done something like this earlier. Now, in the last 6 years or so that we've been working on this project, we released the first version in June 2009 at the American Society for Mass Spectrometry meeting. And to be honest, we were really shocked when we had hit over 100 installations in a month. And now we're routinely at the amount of 600 to 800 installations per month. And all in all, we've had over 30,000 total Skyline installations. So this is definitely was a huge need within our field. And it's not just that people are downloading it, people are also using it. So every time the software is booted up on someone's computer, it pings our server to see if there's a new version, and we just count those pings. We hit in this last week or so, we had first time ever 11,000 startups or boot ups per week. So this was something that's, again, this is definitely being used a lot. And we can also check to make sure that people are using the newest versions, etc. Okay, so I talked a little bit about, we built software, but you need to measure this one individual gene product or this individual protein. How do you decide which peptides you want to measure for this targeted analysis? It's not a simple problem. So a lot of other labs have started to say, well, we should use a lot of our DDA discovery data for this. And so there's been these repositories that people have been building, like Peptide Atlas is one, the Institute for Systems Biology. And if you looked at a protein like ApoA1, which is a very important protein in HDL, so high density lipoprotein, the good cholesterol, if you look at the data within Peptide Atlas, it's pretty good. It's all the peptides that are shown in red are the ones that people have found previously in other experiments. You notice that the ones that are missing are actually from the signal peptides, so it's good that they're not present in Peptide Atlas. And we can rank them based on those things, based on a number of different scores. The downside is if we actually measured every single tryptopeptide by targeted proteomics, they don't actually correlate that well. So if you were to choose the best ones based on a repository, you'd choose these ones out here. But if you really want to measure the best performing peptides, you'd choose these three peptides that are shown in red with the most intense signals. So that was definitely shocking to us. So basically, we can't use these existing repositories very well, and maybe it's just better off that we measure every single peptide. So it's fairly straightforward for a 30 kD protein like ApoA1, but how about a protein that's a little bit more complicated like ApoB? And so ApoB is the main protein in LDL, or low density lipoprotein, the bad cholesterol. And it's an enormous protein. It's over 500 kD. It's a very complicated protein. It's not that easy to deal with biochemically. And but we did measure every single peptide by mass spectrometry. And we compared that again with the number of spectra that people were getting back from DDA runs. And what was shocking is that if you rank them in red, here is shown is the peptides based on our targeted experiment, the most intense ones, the least intense, and the ones that are sampled most frequently by data-dependent acquisition are shown in purple. So you wouldn't even pick a peptide that were in the top 100 if you were actually measuring them entirely by using the spectrum counts that are present in different repositories. And this is, so it may be actually worth actually measuring every single peptide in a targeted way to empirically determine which peptides give the best response. So people, of course, develop predictors. So why not use these predictions, since there's been about 5 or 6 papers out there on predictions. The downside is all those predictions the predictors are trained using these same repositories. So they're a very good predictor whether or not you'll sample something by data-dependent acquisition, but not necessarily that the peptide is best measure, quantitative measure of an individual peptide by targeted proteomics. So again, if you look here in the red, here's our individual peptide measure signal by targeted proteomics. And if you show in blue, this is the different predictive scores. And you can actually make scatter plots showing basically there's no correlation, or at least a very poor correlation of one. And you could probably say, well, maybe I'm cherry picking them, but this basically doesn't matter which protein you pick. Almost every protein has a similar sort of lack of correlation between the prediction algorithms and the actual targeted proteomics data. So I'll tell you a little bit about how we decided to go about doing this. So we wanted to identify plasma proteins with altered protein abundance in Dahl salt sensitive rat model. So we started off with 7 rats on a low salt diet and 7 that had a high salt diet. These 7 Dahl salt sensitive rats end up developing severe hypertension, getting left ventricular hypertrophy, and congestive heart failure. And we wanted to then go through the literature and to be able to pull out about 109 proteins that were predicted to be involved in or be predictive of either hypertension, congestive heart failure, heart disease, inflammation, etc. that can be measured in plasma. Again, it's like the equivalent of doing our Google search prior to going and visiting a foreign city. And in there, we had Skyline say, predict all the basic tryptic peptides within some given rules, and tell me all the fragment ions that could be used for that. We then had Skyline output this into a single or a set of methods. It was 151 injections, which were measured over about 7 days. So the idea is, instead of actually going and trying to predict what best signals were, best peptides and best fragment ions were going to be used for an individual protein, we were just going to empirically measure all of them. And it just basically needed to be done once, and it only took about 7 days of instrument time. When you consider this consideration of cost and expense and time of making a monoclonal antibody, it's substantially cheaper and faster. So let me just tell you a little bit about what happened. So we did, 109 proteins, 2,100 peptides. There was 151 injections. We then were able to detect 49 of these 109 proteins, 135 peptides, and 780 transitions. We then got this into a single injection, which we call scheduled. So we just measure the actual individual peptide for a narrow period of time around where a peptide comes out. And then the ultimate data in the end, it looks like this. So we start with 109 proteins. We inject 49, 135 peptides. And this is in this heat map is basically those peptides. So these are the 130. Each row is one of the 135 peptides. We can measure 135 peptides in the 14 animals, and each column is each of the 14 animals. The ones on the left are those that were from the healthy animals. The ones on the right were those with the congestive heart failure or the dull salt sensitive rats that were given a high salt diet. And each of them were measured in technical triplicates. So each column is and each row is the mean of the triplicate injections. And there were 34 peptides mapping back to 13 gene products that were statistically different between the groups. But as you can actually see, from the clustering that's done here, it was very easy to differentiate between these two groups. And obviously, the differences here now are just largely the difference between biological organisms. And if we had more more animals, we'd probably be able to find a greater percentage of these individual peptides that were statistically different. And this isn't that surprising. These peptides aren't saying that it's specific for hypertension or congestive heart failure. It's just saying that these peptides are different between these two conditions. And so they could actually just be saying that these animals are sick, but it's actually a good starting point. It's not surprising that we find such a high percentage of our total peptides that are different between them, because we started with a bunch of knowledge, again, based on what was in the literature of things that people had proposed actually may be useful to differentiate between congestive heart failure or hypertension or inflammation or cardiovascular disease. So what are some additional examples that we try to use? And so one thing that we often do in our department here is, of course, RNAi, and we're a genomics department, and what people will often do is they'll try to do large screens with RNAi knockdown, and then then they see which of these knockdowns actually have a phenotype. But there's, of course, lots of cases where you don't actually get knockdown on the target protein. So we wanted to be able to have a very rapid way in which someone could actually screen for whether or not an actual individual gene product was actually knocked down or not, without actually having to have an antibody. Because again, a lot of model systems aren't good antibodies. So here's just two examples. These are two gene products, organ binding peptides in Drosophila. And we developed an assay between the two of them. The peptides shown in red are the ones that we initially screened for. We were able to use this. So basically, this B column right here are the measurement of an individual peptide that is a control, sort of basically the equivalent of like a loading control. And then the peptide up here is the peptide that's actually being knocked down. And in this case, we're getting greater than 99% depletion. On the right-hand side are the exact same samples done by Western blot. We confirm that the target protein that was a control has this very similar ratio to what we're getting based on the mass spectrometry data. And the interesting thing here is that the Western blot by ECL shows there's no detectable amount by ECL of the ACP70A. So it shows but that's largely just because the dynamic range of the ECL is fairly limited to about 10. But the dynamic range of the mass spectrometer we already talked about was about 10 to the fourth. So even though this peptide is knocked down by greater than 99%, it's still easily and readily detectable by mass spectrometry. It's just we can actually truly measure that it's been knocked down by greater than 99%. We did this for several other cases too, and of course, just like you can have an antibody to multiple different domains for gene product, you can measure multiple different peptides to provide you different conformation, maybe different isoforms or splice variants, etc. So here we measured 2 different peptides for this individual gene product, which provide independent confirmation of knockdown. And again, we had a different loading control. We're not limited to fly proteins. Here's an example showing p53 being knocked down, and the results that we get by mass spectrometry and completely unfractionated sample tend to reflect very closely what we actually get by Western blot. And we use CapDH here in this case as our loading control. Again, just a very simple example. We have the mass spectrometry equivalent of a Western blot. We can do this for multiple things in parallel, and we don't have to make an antibody or any immunoaffinity reagents in the process of doing this. And we have lots of ways in which we check the specificity, but mostly it's by synthesizing synthetic peptides or by actually using recombinant proteins. Okay, I told you a fair amount of information now about where we actually refine the data in the actual sample. But what about for very low abundance proteins like things like transcription factors? So if in this case, it would help if we could identify which transcription factors we have seen before and the fragmentation figment of these peptides. But so I talked about the challenges of using existing databases, but this is even a more extreme case for something like transcription factors, because if you consider the transcription factors that we have encoded in the human genome and what fraction we even have any data in any public repositories, majority of them have no tryptic peptides or no peptides at all that have ever been seen by the public. So of like something 760, I think, transcription factors that we were looking for, trying to build assays for, more than 600 of them had no peptides found in a NIST spectrum library at the time that we started doing these experiments. And then of the remaining ones, some of them we had 1 or 2. So a majority of them, of the proteins that we would be interested in, there would be no information even to go look for in the first place in the public repository. So we decided to kind of base our approach on how we were going to do this was to be able to make proteins recombinantly. I saw a seminar by Josh Labar at Arizona State University where he had been talking about making thousands of proteins on a protein microarray in parallel, and I mean, well, why don't we just do this in 96-well plates? Had a lot of communication with him about it, and it turns out, what we were able to do is use his cDNA clones for making individual proteins recombinantly using an in vitro transcription translation system that we worked on with Pierce, and this was based on the HeLa cell extract. And all of these cDNA clones are fused with GST, so we started off with 96, but we eventually did this for 730. We did in vitro transcription translation. We enriched for the proteins by glutathione resin. We digested with proteins, and we monitored all three peptides using selective reaction monitoring. We confirmed things by silver stain gels and also by Western blot. But basically, the data looks like this. For an individual gene product, we now have intensities for individual peptides. We know exactly where they elute from. We know which fragments contribute the most to what portions of the signal. And in most cases, the proteins that we produce are relatively pure. There happens to be in our extract a couple of glutathione binding proteins, but our proteins are still more than half the target proteins that we're making. All the proteins are fused to GST, so we were able to use peptides from GST to quantify the amount of the produced gene product. And there's not a lot of proteins that are actually produced, but still there's plenty in order to measure by mass spectrometry to work out this assay development. We had a 100% success rate in expressing 730 proteins. There were still several proteins that were relatively small and didn't produce good tryptic peptides. So in those cases, even doing a targeted quant assay by mass spectrometry would be hard in the first place. And it's good to actually start by knowing that, as opposed to just not knowing what you're missing. We measured some of these things in the in vitro synthesized proteins. We also measured them in the nuclear extract. And while the data from the nuclear extract was a lot noisier and had a substantial more amount of interference, these peptides eluted the same retention time, had a similar fragmentation pattern. We can get a lot of information about like the tissue-specific expression of individual proteins like GATA2. So the data, using CTCF, we also see, it's more abundant in FG2, and so we get, right off the bat, without too much work, we get a lot of quantitative data, or we can get a lot of quantitative data between different conditions. We now know what we're looking for. We know what signals and what data that's out of the noise actually represent our target peptides. And we do have a lot of sensitivity. We feel pretty comfortable now that we have absolute structural specificity, because we have not only the hydrophobicity of the peptide, the mass of the peptide, the mass of the fragment. We also know now the rank order of the individual fragment ions that provide us with a very strong qualitative confirmation that we're measuring what we think we're measuring. So the first applications we did with this were done with John Stavatoyannopoulos' lab. A very talented graduate student, Andrew Sturgachis, started these experiments. So basically, he wanted to know which transcription factors separate into different discrete chromatin compartments. And so we basically took the nucleoplasm, we added different amounts of salt and kept taking the soluble component from there. And so basically what we're doing is displacing different amounts of proteins from the chromatin, and each of these fractions are then measured by mass spectrometry. We can then make different sort of heat map plots like this to measure different transcriptional regulators and see some of these proteins are eluted only in the isotonic buffer. Some are stable at very high salt concentration. Some elute actually kind of an intermediate salt concentration, which indicate that it's more loosely bound than those that are very stable. And some only come off at when you add a DNase or an enzyme like an MNase in order to completely chop up the DNA into its components, and like the histone proteins. And then, of course, things like the nuclear membrane, even when you chop up the DNA, it still remains in the pellet. Andrew wanted to do this to even greater scale, so he extended the number of fractions, he extended the number of proteins to more than 300 proteins, and he was able to get a similar sort of pattern or heat map to show basically the chromatin segregation of these 300 transcription factors in a very simple, quick analysis. He was able to do this for multiple different cell types and do it very, very rapidly. We confirmed a lot of this data by Western blots, and it's pretty some exciting results. So when we started this experiment, one of the proteins we measured was Cap1 isoforms, and we measured these 3 different peptides, and in general, these 3 peptides all agreed across the different samples that we measured, with the exception of one sample. There's just one sample right here, the 600 millimolar stable sample from the K562 cell line. We noticed that this one peptide, this VLVNDAQK, had a much lower abundance than the other 2 peptides. And in most cases, people would just chalk this up and say that, well, this is an outlier, we'll throw this out. This really began to bug Andrew and us, and we try to track this down. Well, when we spent a little bit more time looking at this, it turns out there's actually a 3rd isoform, and this 3rd isoform does not include this extra 3rd peptide. So by measuring different individual peptides, you can actually get different isoform-specific type information. So this purple and blue peptides represent the signal from 1, 2, and 3, and the green or the yellow peptide represents the signal from this isoform 1 and 2. And of course, this is tissue specific, because in HepG2 we don't see this difference at all. We confirmed this by Western blot using an antibody to the carboxy termini, and you can see here there is a smaller molecular isoform that's present here. Okay, so there's still a challenge here in the fact that these assays still take a lot of work. They take a lot of time and effort to set up. And what would often happen is you'd measure an assay measuring 100 peptides or 100 proteins. You'd measure it in 1,000 different samples. And then you'd go, geez, there's this paper that came out, and in this paper it says this other protein, this other peptide is actually really important in this sample set. So then you have to think about going back and remeasuring all those samples again. So what we really wanted was methodology that had the selectivity of targeted SRM with the breadth of data-dependent acquisition. So we wanted something that was going to be unbiased, but we wanted to be able to make it so that you could target very specific peptides computationally. And if you need to go back and add an individual peptide later, you don't have to go back and rerun the experiment. So basically, the Aerosol lab has been calling this sort of basically a digital molecular archive of all samples, and other people begin to call this these same sort of things. So I've already talked a little bit about the fact that there's these alternatives. There's this data-dependent acquisition, and then there's also the selective reaction monitoring, or sometimes parallel reaction monitoring as a term that's used using a slightly different type of instrument hardware. It basically targets these very narrow mass charge windows. It doesn't cover the entire mass range, only covering the selected region of the entire mass range. You have this, the nice thing about it is that you have this repeated duty cycle, so you can actually generate these extracted ion chromatograms and use that to quantify individual peptides. But the peptides that fall in the in-betweens in this precursor mass charge region really are a challenge because we overlook them. We don't have an ability to provide any other peptide quantitative information for peptides that fall in these missing regions. So there's this new data. It's not too new. It's about 10 years old as a data acquisition strategy called data independent acquisition. And the idea and the concept is that we just open the mass range up. And we fragment everything in that window, and then we hope that the fragment ions have different masses. The data looks a little different. So basically, if we take this 500 to 510, we get data that looks like this. And so you have these individual fragment ions that covary versus time, and you can actually make plots of an individual spectrum at any point in time. And again, you can make a slice of the data in the other direction. And this now, instead of being just the mass of the peptide, is the mass of the precursor on the individual product time data. It's very similar to what we get with a triple quad type data. We can extract the data after the fact. So if we have an individual peptide that falls in this individual mass window, we can extract then the fragment ions from those that were predicted from the individual target peptide from within that window. And we can plot it in this sort of way. Here's just plotting it in 3 dimensions. Here's plotting it in 2 dimensions like our software does. And we can also do it for the precursor information and the fragment ion information, and both of them provide sort of different pros and cons to the interpretation of total data. So one of the challenges that we have that's kind of not appreciated with this type of data is the selectivity. So if you wanted to measure this individual peptide in a yeast lysate with only 10 mass charge wide windows, and a lot of these experiments are done with more wider windows, like 20 or 25, this is what the data looks like, and you wouldn't know to really say, pick this individual region of time here and say this belongs to our target peptide. Sort of like doing a Western blot with a very non-specific antibody. We've now, because we've opened up the window of the precursor mass, there's lots of things that fall in the mass. There's lots of things that have those sort of fragment ion masses within a given range. So as we open up the windows from being narrow, our spectra looks kind of like this, and now I'm plotting with mass to charge. And it gets to the point where as you open your window quite wide, your target peptides are actually not necessarily the most intense peptide within the isolation window that you're interested in. My students also like to say that data-independent acquisition is about balance, right? So we ideally want to have smaller windows, but in order to do that, we need to sample more frequently, which means we don't sample the chromatographic peak very often. And likewise, if we want to go to something like 5 mass charge wide windows, we would sample the peak not very well at all and wouldn't be able to get very good quantitative data across the chromatographic peak. So one option, of course, is to make multiple injections, and then that increases the frequency, which you sample the peak, and it has high selectivity. But the downside here is that you now have you've decreased your throughput by a factor of 2 because you have to make multiple injections to cover the entire mass range. So we began to think about this a lot over the last 3 or 4 years. And one of the things that was an eye-opener to us was when this new instrument, the Q-Exactive, was released on the market by Thermo Fisher. It has some interesting combinations of hardware. It has a quad, so we can isolate an individual mass. We could subject it to fragments, and we could store those fragments. And then we can measure those fragments in the multi-track mass analyzer. And this is done generally in series. And the interesting thing is that the time it takes to fill that C-trap is done in parallel with the previous acquisition. And that fill in general is relatively short compared to the acquisition of the mass spectrum. So what we wanted to do was to fill multiple different disjoint windows and collect a mixed spectrum with the intention that we'd have a computational strategy to allow us to demix or demultiplex the data at the end. So bear with me a second while I try to describe this a little bit. Imagine you wanted to measure 4 mass charge wide windows that span from 400 to 800. You'd have 100 4 mass charge wide windows. So let's say we wanted to randomly select 5 of those windows. We could take those and measure those, and we can isolate them all in series. We could fragment them. We could store them in the trap and then measure one spectrum that contains all 5 of those separate 4 mass charge wide windows. We can then take 5 of the remaining 95 that are left and do the same thing again. And we keep doing this for the third scan, etc., until we get to scan 20, where there's only 5 windows left. We select those and then measure them. Now this is where it gets a little interesting. In the 21st scan, we refill the list and start the whole process again. So we randomly selected the 21st scan, 5 more scans. And there's a part here that's pretty cool, and that is that between scan 20 and 21, there's a single mass window that's shared between the two. The remaining 4 ones are different. Because of this, if there are peaks or fragments in the same isolation window that are similar between scan 20 and 21, they must come from the same isolation window. Those that are different must come from the other ones. So the idea here is, can we take this mixed spectrum that contains 5 combinations of 5 separate windows and ultimately demultiplex them into the 5 individual components? And I'll spare you the linear algebra and the math behind here, but basically, yes, we can. Here's the data here that's shown measuring 20 mass charge worth of data in each individual scan. Here's the blow up of the data. And when you demultiplex it to the individual 4 mass charge wide isolation window, the data is much cleaner, much easier to assign the correct peptide location, and the quantitative data is much cleaner and better signal noise. We have multiple different examples of this. Here's this 10 mass charge worth data independent acquisition data. Here's 20 mass charge worth of what we call MSX, this multiplexing data, and demultiplex to the individual components. And when you blow them up, you can obviously tell that the signal and noise are much better. There's a lot less interference. So again, it's like having an antibody with much better selectivity. Instead of having an antibody that has 10 mass charge with the selectivity, we actually have it down to 4 mass charge, more than double, even though we're covering 20 mass charge with the data in each individual spectrum. Again, here's just a third example. It's definitely very powerful technology, and we're trying to make bigger improvements of it. The final kind of catch here is that, yes, it is also quantitative. This was something we spent a lot of time validating, and depending on the complexity of the mixture, it either is or is not more sensitive than using just the data by using the more traditional data independent acquisition. One thing that was it's a little bit striking here is that here is an oxidized form of the same individual peptide. When you have 10 mass charge wide worth of data, that oxidation is a 16 Dalton difference. It's 8 mass charge, so both of them fall within the same isolation window. They have almost the exact same fragmentite up to the modification, so an oxidized methionine. So you can't really distinguish between it until you get down to that 4 mass charge isolation window. So this bottom chromatogram is actually 20 mass charge worth of data that demultiplies the individual components. So there's a couple challenges here. One is the wide precursor windows fragment multiple peptides at once. We can solve this using this MSX approach, this multiplexing approach. There can be some limiting factors, and that's largely due to sensitivity. And that is that it takes longer to fill the ion trap in order to do this. And also, not all instruments are really capable of doing this MSX experiment. So we wanted to modify the experiment design in order to improve this a bit. So if you had a 20 mass charge worth of data and you had 2 individual peptides that span that isolation windows, what we'd want to do is, you could shrink the windows down to 10 mass of charge and cover half the mass range, and you'd improve the specificity, but you'd miss things from 500 to 600, 800, 900, or 200 mass of charge that are outside the window. So one thing that we came up with was the idea of is shifting the windows. So just to kind of show this like this. So now what you end up happening is you get some peptides that fall within the same isolation window. They would look like this if they were not shifting. If you shift the window, those that are in the other half of the window form the sawtooth pattern, and we can ultimately demultiplex this to the appropriate components. And just to kind of give you a sense is that if we measure an MS1 data versus doing 10 mass of charge with the isolation, which is half the mass range, so less comprehensive, versus doing 20 mass of charge data that's demultiplexed, we're almost getting the same sort of sensitivity as the 10 mass of charge data using 20 mass of charge worth of data and then demultiplexing them. And it depends, of course, depending on how many fragment ions that you actually are using. But regardless, when you have 20 mass charge worth of data, your lower limit of quantitation, your sensitivity and your quantitative capabilities are not anywhere near as good as if you use narrower windows. But with the narrower windows, you have less comprehensive data analysis. And so what we can do is we can shift those windows and then demultiplex them computationally. And our software tool, Skyline, handles all of these things automatically in a seamless way to the user. So there is still some issues here. I made this sound fairly straightforward. We have software tools that can manage this. There is a dirty little secret in doing these analyses, and that is when you try to measure these things by data independent acquisition, we have a big difference in simple things, just like file size. So by selective reaction monitoring, where we target individual peptides, so basically we have gaps in our mass range, but we measure selected peptides very sensitively. I showed you with the transcription factors. The file sizes are tiny. They're only about 5 to 20 megabyte a file. So it takes a few seconds just to import the data into our software tools. With data independent acquisition, the file sizes are anywhere from 200 megabytes, so easily a factor of 10, if not a factor of 40 or 50 size. So we're looking at minimum 30 seconds to tens of minutes to import this data into our software tools. So if you consider something like 50 runs, which of course would be pretty much the minimal quantitative experiment we want to do, we're going now from what used to take us 10 minutes to import the data into a software tool to hours to import the tool. So we began to start thinking about ways to solve this, and we have another project that's being done jointly between our lab at the University of Washington, Nathan Yates' lab at the University of Pittsburgh, InfoClinica, and also it's all being done on Amazon Web Services. We call it this courseproject.org. And basically, it was meant to be a platform for storing and managing and sharing mass spectrometry data. And it's based in the cloud, and there is some pros and cons of cloud computing. So one, of course, is the massively scalable storage and compute resources. You don't need to worry about the fact that you're running out of disk space all the time. You just need to pay for more storage at Amazon as you need it. You can achieve economies of scale. So basically, if everybody in the entire community is putting things into the same account, the price that is costing the community, we get a benefit because Amazon Web Services, of course, will now charge us less money to have, petabytes of data than we would if we just had a few terabytes of data. We only pay for what you use. If we buy 20 terabytes of storage today, most of that storage is empty until 3, 4, 5 years down the road when we finally filled up that storage. So we've been paying a lot for disks that are largely empty. We can access that data anywhere in the world. If you use a good cloud computing service, there's a lot of redundancy and security, and there's a lot of just also improved flexibility. So let's say you built a lot of compute hardware in your lab, and in a month's time you realize you needed machines that had more RAM, random access memory. The only way to solve that problem, would be to actually go and order new computers with greater amounts of RAM. But if you use the cloud, you can just change the instances that you use to using higher memory instances. So it's actually great that you can actually change directions pretty quickly. There are a lot of criticisms, though, of course, using cloud computing. There's there's a lot of ownership of the infrastructure in tight federal funding times. If someone lost their funding and you are leasing the computers, you then basically now lose your computing infrastructure. When if you bought the hardware, you actually have, even if they're older computers, you still have some computing hardware. You now get stuck with a monthly bill. It's sort of like, again, leasing versus owning a car, or renting a house versus paying, purchasing a house. A lot of the software applications require modification, and one thing that's a kind of thing that we actually haven't found to be a big deal yet, and I'll describe this a little bit, is limited data transfer rate. So some universities may actually have an issue, or institutions may have an issue where the bandwidth of the internet may be a bottleneck. And if we're talking about collecting data files of about 1 gigabyte per hour, it's not really a challenge to upload it a gigabyte per hour. So we can upload it in parallel with the data collection. So about 15 months ago, we released this initial course project. It has a user interface for uploading, sharing, and making mass spectrometry data public. We now have quite a bit over 800 users, about 11 terabytes. This is a little bit outdated. The data files are stored in a very unique distributed data structure that allow us to instantaneously access individual raw data using a sort of massively parallel compute infrastructure. We have a lot of basic chromatogram and spectrum viewers. And now, and this is just showing what it looks like. On the right-hand side is Chorus, on the left-hand side is what we modeled it off of, basically Google Drive, and on the left-hand side is basically seeing, individual files and multiple people are accessing, and you can put them in directories and share them, and that's basically the same thing that we're treating mass spectrometry data is a hosted things. You can have data stored in projects and experiments of that project, and you can visualize them, analyze them, etc. We've built viewers. You can look at the mass spectrometry data, and you can also begin to analyze it. This will be available starting in the next couple of months. The ability to do things like database searching. Like I mentioned, that the technology called Sequest, it's basically been implemented on the cloud. You can set parameters and kick off analyses. You can visualize the results, looking at proteins, individual peptides within your data, and you can start to look at quantitative analyses, et cetera, comparisons between lots and lots. We can align chromatograms, et cetera, and sort of calculate statistics. What used to take hours of compute time now is done in a matter of minutes. Okay, I mentioned earlier that one of the challenges with data independent acquisition, and the dirty little secret, is just that it takes so long to extract the data from these files. So what we wanted to do is to be able to store all the data in Chorus, so someone didn't have to carry around a terabyte on their hard drive. On your laptop, imagine a lot of laptop hard drives aren't that big to begin with. And a lot of laptops are actually pretty pokey too. So we wanted basically the client to be able to make requests for chromatograms to the cloud and Chorus to be able to return this. And we weren't sure how fast or quick this could be. And I have a quick sort of video to kind of illustrate this. So here we are looking at a laptop. It's a fairly fast laptop. It's an i7, 8 gig RAM with an SSD. We have a local disk versus that from the cloud. And if we were to copy these things from the internet, there's about 15 gigabytes of data that's stored, remotely. And this, of course, isn't simple to be able to download. It ends up taking about an hour and a half to download in full. And so if we import it locally from the local disk, now that we've downloaded it, took an hour and a half, there's importing 6 different files into Skyline from the local SSD, and we're limited now by the hard disk, the CPU infrastructure of the laptop, versus from the cloud. So we can go and import the results from the cloud. We can now browse the cloud just like you can versus using Google Docs or Google Drive or Dropbox. You can browse this directory structure remotely. We can import those 6 files remotely across the internet. And you'll see that because of the way we store and manage the data on the internet, on the left-hand side is the data being imported. 1 of 6 files has already been uploaded into the software through the internet with very little bandwidth problems. On the right-hand side, we're reading it locally from disk, which you would expect would be much faster because there's no throttling, of course, of the internet. And so what we found is that because of the data structure that we can store the data in, and that we can have multiple CPUs reading the data in parallel, we can essentially stream the data locally down to the computer. It's much faster to read it remotely across the internet than it is to read it locally on an SSD drive. So now, basically all 6 of the files have been loaded into the file on the left-hand side. We're still only loaded 1 data file from disk. And now you can go and start already looking at the data from the cloud. You can start to look at all the individual components. You can start to analyze the data. You can look at spectra, which again, it brings it from the internet. It's extremely fast, very low latency. Again, reading it through remotely across the internet. It's just something that's to come. Okay, a little bit in conclusions. I really think targeted proteomics is going to be a major growing area of proteomics. The idea of performing mass spec equivalent of a Western blot is really powerful. Modern hardware and novel data analysis strategies are beginning to make data independent acquisition a realistic solution. While data independent acquisition has existed for a decade, it really hasn't become feasible or practical until the last 3 or 4 years. Method setup is much simpler for data independent acquisition, although the downside is that the data file sizes are huge. If you don't actually need the data for the other peptides, it's probably still better to use traditional targeted measurement. We're being limited only to peptides that have been seen previously is not only impractical, but won't use targeted proteomics to its full potential. So we're advocating that you don't use things like from a repository to target those individual peptides. They actually make the peptide recombinantly, build assays very specifically to the individual proteins, etc. I do think that the future of proteomics will require that our data will need to be stored in the cloud and accessed remotely, and those features are coming in January. There's been a lot of people who've done a lot of work in this. So a lot of our targeted proteomics work now is being done by Jim Bollinger. A lot of our data independent acquisition is being done by Jared Egertson and Rich Johnson and Sonia Ting. Brendan McLean and Nick Schulman are software engineers that made a lot of these tools possible and fast and robust. And all the course development is being done by a not-for-profit we set up called Stratus Biosciences. And a lot of the quantitative assays we're building are being done, at least in consultation, collaboration with Andy Hostengel's lab and Clark Henderson in his lab. We want to make sure ultimately that the quantitative measurements we're making could actually be transferred to the clinic down the road, just because there's of course a huge need for that. And thank you very much. I'm happy to answer any questions. So there currently aren't any questions at this point. I'm happy to answer any. I have some. There's a lot of information covered in a short period of time. So thank you very much for attending. I really appreciate the opportunity to be able to tell you a little bit about things we're trying to work on, and hope I was able to tell you a little bit about the challenges and also the strengths of doing targeted proteomics. Thanks.